So a very warm welcome to all of you guys. Today, we are going to discuss a topic regarding genome data analysis. So as we all know that genome data is one of the important approach in today's era, where it is dealing with respect to the big data analysis for the biological data sets. So whenever we want to discuss an biological data sets with respect to the comprehensive analysis, so we need to perform a genome data analysis. So basically, that is what we are going to discuss in today's session. So welcome all of you. Myself, Dr. Nilofer. I'm a PhD scholar. So today we are going to discuss the same topic ahead. So as an introduction, so basically a genome data analysis will provide you a comprehensive guide to uncover the genetic code of a molecular biology. So in this presentation, we are going to explore the specific genetic code to analyze the biological big data with respect to the genomics approach and what exactly the applications are available in the modern research with respect to the research and application. So genetic code, as in basic, we have to understand the nucleotide sequence and even the codons, which are ultimately the fundamentals of decoding the genetic information. So basically, in that specific gene, we are going to explore the certain kind of mutations which are related with the specific disease and how we can uncover those mutations with respect to the identification of most potential applications, such as the biomarker identifications, and also with respect to the potential drug targets identification. So for that particular purpose, as we all know that genome sequence techniques is playing very significant role in today's era. So before that, we do have a Sanger sequencing, but now we are using a next generation high throughput technique such as NGS technique. So basically in the NGS technology, we used to process the particular sample of the sequence data against the reference genome to interpret the particular mutations with respect to the novel variants. So here, basically the bioinformatics tools will help us to analyze and also process the biological big data, even using the omics approaches such as the genomics. So the basic tools such as the BLAST database, which stands for the Basic Local Alignment Search Tool, GeneBank, which provide you to analyze your biological data and even interpret the results with the genomic context. Apart from this, we do have certain a standard or else the target-oriented databases and even the resources where it will help you as well as guide you to perform your genome analysis against reference genome. Whereas the genetic variation analysis is ultimately a motto or else the applications to perform a genome analysis, such as to understand the SNPs. There's nothing but the type of mutations. So SNP stands for the single nucleotide polymorphism, CNV, that is a copy number of variations, and even the structure variants. So if you identify those mutations against the reference genome with respect to the target of your interest, so ultimately, guys, you are uncovering the informations of genetic diversity with respect to the disease susceptibility. So this informations will ultimately help you to explore the certain informations with the genomic context. So we do have certain applications such as the functional genomics. So as you all can see that, guys, that genomic application with respect to the functional interpretation of the biological data will ultimately help you to explore the gene expressions patterns and even to understand the regulatory elements which provides the insights in the functional aspects of genome data. So exactly this functional genomics data sets will give you a significance finding and to understand the biological significance with respect to the biological process, such as the molecular pathways, biological process, and even the cellular mechanism. So I hope that all of you are getting, right? So the overall applications will ultimately leads with the clinical applications, such as in future, we are trying to identify the precision medicine and even to diagnose better techniques with respect to the patient diagnosis. So this is ultimately a broad application of working with the genomics data. So we all have to even know which are the current applications and even the future applications for so. So there is also some ethical considerations are there to working with respect to search clinical data sets to design certain kind of potential drug targets or else the precision medicine whenever we are integrating some clinical data, right? So genetic testing privacy concerns are the critical in the genomics data analysis. And remember guys that we can overcome those things using the bioinformatics platform. So ultimately, we are uncovering the information of the genetic informations with respect to the genome analysis. So whereas what are the future perspective of designing certain genome data analysis is ultimately we can take the advancements of the current as well as the future-based high throughput techniques such as the single cell sequencing. CRISPR-Cas9 technology is ultimately providing a shape for the future for the genome data analysis. So this potential future will development and even the emerging trends will give a better understanding about the genetic code analysis. Whereas the overall conclusion is that the unraveling the particular genetic codes through the comprehensive genome data analysis will give you an advanced scientific research as well as the medical breakthrough. So ultimately, this presentation has provided you all a true understanding of the genetic codes and even to analyze its by far-reaching its implementations. So I hope that you all of you get a better understanding about what is exactly mean by a genome analysis and what are the future applications with respect to the bioinformatics tools and their application. So thank you all of you.